Okay, good morning everyone and welcome to the third webinar in the Human Relevant Research in India series, which we have been having for the past 3 months. And so I'll just take like 1 or 2 minutes of your time and just share just 1 just 1 minute, 1 second. Okay, so is the slide visible?
Yes.
Okay, so I'll just take like 1 or 2 minutes and just talk about this initiative. So I am Surat and I am a Senior Research Associate with the Center for Predictive Human Model Systems, which was established at Atal Incubation Centre, and is a collaborative venture between Atal Incubation Centre CCMB and Humane Society International India. And this center was established last year as a think tank to enable more research in India that is relevant to humans. Like for example, currently predominantly research is based on animal models, but however, it is increasingly emerging that a lot of research in animals is not really translatable to humans. And so we need, and now that we have in the past few decades, we have developed a lot of new methodologies in both in terms of new technologies and also computational tools, which can now be harnessed to make science more relevant to humans. And so to do this, so what do we do to enable to enable more of this research in India? So for the past 1 year, some of the activities that we have been doing is first write white papers on various of these identified areas, such as new methodologies, such as microphysiological systems, which includes organoids and organ-on-chip, and then adverse outcome pathways and systems and computational biology, to understand what is the status of research in India in these areas, and to understand also the status quo of funding and what are the regulatory limitations to do more of this research in India. And the main aim is to identify where the problem areas are, so that we can start having a dialogue with different stakeholders to address those problems. And we've already had like a few workshops, and we also plan to have more workshops and webinars to increase awareness amongst both early career researchers and also students to create awareness, so that these are the methodologies that are there and they can use these methods. And we've also been involved in writing few reviews which are currently in review. So yeah, and popular science articles. So again, to the point, for basically to address to increase more awareness. And we've also given out a grant in the past year which is currently an ongoing thing. We've already selected 2 people to work on adverse outcome pathways, and Dr. Gayan, he is also one of the recipients of the adverse outcome pathway grant. And so this webinar series is also part of this initiative. And with that, I would like to end and just introduce Dr. Karthik, who would be our first speaker. And Dr. Karthik did his PhD in systems biology from IIC Bangalore, and then his postdoc from University of Zurich before joining IIT Madras as faculty. He is also the coordinator for Initiative of Biological Systems Engineering at IIT Madras. And currently his lab works on computational tools to understand, predict, and manipulate complex biological networks. So with that, I would like to ask Dr. Karthik to begin his presentation. Thank you.
Hi. So good morning, everyone. It's very nice to get online and during these times, and still science goes on as usual. So I look forward to a very interactive seminar. So please type in your questions as they arise in your head on the chat box. I'll try to keep an eye on the chat box and try to answer them as I go on. And of course, we'll have some dedicated time for discussion at the end, I presume. So once again, I'd like to thank the organizers for giving me this opportunity, and let me just get going. I'm just going to talk to you about computational approaches to understand biological networks. I'm going to talk mostly about graph theory. Our lab does a lot of work in constraint-based modeling, but today I'm going to talk to you more about graph theoretical approaches. Today we have a huge amount of quantitative information on biological systems, and it's quite a challenge to. Are you able to hear me?
Yeah. We can hear you.
Okay, it says the host muted you, unmuted you, and stuff. So, okay.
No, we can hear you.
Yeah. Okay, so how do we make sense of this information? So one way to go about is to build models and networks to organize and assimilate this huge amount of genomic, proteomic, transcriptomic data that has come our way, metagenomic data, if you will. And models are, of course, useful to ask various questions about complex systems. I think most of you in the audience are probably in agreement with that. And a good model can often answer questions that may be very difficult to address in the wet lab. And I'd like to put in two very nice quotes here. One from the Nobel citation in 2013, where they say the computer is as important a tool for chemists as the test tube. And you can score out chemists there and say biologists or any other field today. And I think, we've all seen the importance of computational biology in the recent years. And then one of the greatest computer scientists, Donald Knuth, who basically said that he can't be as confident about computer science as he can be about biology, which has so many years of exciting problems to work on. So there are different types of networks in biology, right from metabolic networks that you see here, to protein interaction networks and so on. There are gene regulatory signaling networks, food webs, if you will, and so on. And you can even look at protein structure or molecular networks. And I will, for good measure, talk about that at some point in the first problem that I'm going to discuss today. But if you see, the cellular function itself is a result of all these underlying networks. So this is the outline of my talk. I'm going to give you a brief overview of our lab, then go into three different topics, but two of them I'm going to touch upon very briefly and focus a lot on how do we go about identifying interactions in microbial communities. So this is our lab, and we are an interdisciplinary group. We focus on building scalable algorithms for metabolic network analysis and understanding microbial interactions in communities and with applications in metabolic engineering and so on. We also do quite a bit of machine learning and big data analysis with respect to both clinical and omic data that we have from various collaborators. So we are part of the Robert Bosch Center for Data Science and AI, as well as the Initiative for Biological Systems Engineering. So if you see what we work on in our lab, we have a few recurring themes. The tools are basically constraint-based modeling, complex networks, machine learning, high performance computing, and so on that you see at the center of this cartoon. And the applications are in various different areas. We look at metabolic engineering and pathway identification. We look at synthetic lethals. We look at designing regulatory networks. And a lot of our recent effort has gone into understanding interactions between microbes in consortia. And we've also been looking at integrating multi-omic data. So these are some of the key research contributions from our lab. I'm going to talk about 2 or 3 pieces of this work today. But if you see, we work a lot on algorithms, and they're all available from our GitHub website. And we also go on several, the applications are in metabolic engineering and in terms of uncovering or understanding newer biology. So I'll first talk about a very interesting problem. This, I think everybody can appreciate the problem for sure, because it's a classic high school question. So how do you convert the molecule on the left to the molecule on the right through a bunch of reactions, right? So in school, you must have some memory of organic chemistry, and you would say that use some Kolb-Schmidt reaction or a Diels-Alder. You remember all those names that we used to learn in school to convert the molecule on the left to the molecule on the right. But today, of course, you don't have to memorize all these reactions, and instead you can learn this from a database of enzymes or enzymatic reactions and say that I could convert the molecule on the left to the molecule on the right by using a couple of novel enzymes. So what we did was we took about 10,000 plus reactions in publicly available databases and learned a very interesting model out of it. So how did we go about it? Our question was, can we build a fully automated engine to predict pathways between two molecules, relying only on structural information? Because there are already many tools that do this task, but they need something more than structural information. They need, some of them need atom-atom mapping information to understand how, what is the reaction mechanism and so on, and things like that. So our approach basically is quite simplified, in which we are only trying to look at the structure of the molecule. So what did we do? We actually represented every molecule as a graph or a network, if you will, meaning that there are edges and vertices within a molecule, right? So what does that mean? So every atom is now a node and every bond is now an edge. And using that, we performed some very nice math on it. We basically used something called subgraph edit distance to map reactants and products, because you in every reaction of the form A plus B going to C plus D, A actually becomes, say, C or D, and B becomes the other molecule, right? So typically, so glucose plus ATP will give you glucose 6-phosphate, and glucose 6-phosphate actually came from glucose. Figuring this helps us go through a chain of molecules, a chain of transformations, and make predictions. We then identify a reaction center where the action is happening, a reaction signature, which we consider to be essential for a reaction to happen, some sort of neighborhood of the molecule, the reaction center. And then we identify a bunch of reaction rules, which say that if such and such bonds are present, or such and such atoms are present in this configuration, they can be converted to such and such atoms. And we stuck these into a network, which finally reduced the problem of pathfinding to pathway prediction to simply a pathfinding on a network, which is quite an easy algorithm. And we also had some nice heuristics to enable that. So finally, what did we do? We could actually realistically rank pathways from a large pool of alternate pathways, even in something like central carbon metabolism, because you can have like hundreds of ways to convert between a pair of molecules. But what is the most biologically plausible way to do that? And that is something we were able to predict using our method, and it's available on GitHub. So this is one piece of work where we basically used two different types of networks. So first one was a molecule itself was a graph, and then we also built a graph of reaction rules and so on. The next piece is quite different. This is more, standard in terms of studying a protein-protein interaction network. And many of you, at least, any of you who've heard even a basic lecture on network biology might have heard of the lethality centrality hypothesis, meaning, or centrality lethality hypothesis. So more central a particular node is, more likely it is to be lethal. So our question was, can we predict essential genes across organisms simply by virtue of its network position? No other information. We chose not to use sequence information or other biologically useful or other biological information. And what we did was a bunch of simple network features, like your eigenvector centralities and things like that. And what we did was, we could, with just 12 features, although we finally did report a lot more features in the main manuscript, with just 12 features, we could get a very high level of accuracy in predicting essential genes across organisms. And what we did very recently, about a week or two ago, is we built a database out of this, and you can access it here and give us your feedback, which can give you predictions for more than 2,700 species from the string database. So we basically extract the networks out of string, run it through our algorithm, and make predictions of what are the genes that are most likely to be essential in these organisms. So this is a completely different network, right? So first we talked about a molecular network, and now, or a reaction rule network, and now we are talking about a protein interaction network, and how it can be used to make predictions about essentiality using classic machine learning methodologies. So given this, let me cut to the main topic of today's talk. I'm going to talk to you about identifying microbial interactions in communities. So this is not a single organism, but pairs of organisms which interact with each other, and how can you study their potential for cooperation and interaction. And microbes are basically ubiquitous, but they never exist in isolation, right? They always thrive in dynamic environments, and they form microbiomes. Some of them are like good, right, including your gut microbiome and so on, or say the soil microbiome. But some of them, like a biofilm on a medical device, could be a nuisance. But wherever these communities occur, the community structure is dictated by a complex web of interactions. And even in these interactions, it is known that metabolic interactions predominate. So like say metabolic exchanges and so on. So because of this, we can actually build very good metabolic network models to study these interactions. And of course, these consortia are also widely useful in metabolic engineering. And you can even think about manipulating the metabolism or manipulating a consortium to sort of say help the gut recover. So can you think of designing designer probiotics that will help recover a gut from say some sort of disease? It could be a Clostridium difficile infection, or it could be antibiotic administration or whatever. So let's talk about the most interesting microbiome, which is the gut microbiome. You may have read many reports that there are about 10 times the cells, but I think the consensus now is about 3 times. Human cells are outnumbered something like 1:3 by microbial cells in the body, and a lot of them exist in the gut microbiome. This is a very complex ecosystem and has many roles in health and disease. And the composition itself is highly variable depending upon the stage of life, diet, the environment, the exposure to various other agents, particularly antibiotics. And if you antibiotics are a major cause of gut dysbiosis, and there is a huge collateral damage to the gut flora owing to antibiotic ingestion. And there are studies that say that basically even sometimes even several months, even years after an antibiotic dose, an antibiotic course, your gut microbiota still do not recover completely. So how long does this recovery take? It really varies from individual to individual. But what we found was, this is in collaboration with Niranjan Nagarajan in A-Star Singapore, and there they made a very interesting study by looking at a complete metagenomics of the gut, and they identified 21 recovery associated bacteria that could that were found in greater quantities in individuals whose gut would recover faster following antibiotic treatment. And they found a very specific synergy between Bacteroides theta iotomicron and Bifidobacterium adolescentes, which is a very famous human. These are very, I think, familiar gut organisms to anybody who's studied a little bit about the gut microbiome. But the question is, do these organisms work well together? And if so, how do they? So this is the question we set out to answer. And how do we basically understand microbial interactions in these kinds of communities? And I already gave you a hint that we want to focus on metabolic exchanges because they are quite important no matter what the microbiome is. And so can we actually use metabolic models to do this? Turns out yes, and there's like a whole barrage of methods that have been applied. So including constraint-based methods, graph-based methods, population-based methods, and so on. And there are many more methods being developed. But the challenge still is in terms of building accurate models. It turns out that many of these models are not very accurate in terms of stoichiometry and so on. And it's also, there are a lot of differences between the kind of models that are made by different groups in in the community. So can you make these models talk to each other? That's another challenge because the naming conventions and the standards that people adhere to are quite different. So there are these challenges, but there are also very many useful resources through which we can build such models very effectively. And we also have a monograph that talks about in quite a lot of detail about all of these methodologies. So how do we now take a metabolic network, represent it as a graph, and throw some algorithms at it? That's the next question. So there are various ways you can do it. You can think of making a substrate graph. So this is a simple substrate graph, where glucose gets converted to glucose 6-phosphate, then to fructose 6-phosphate, fructose 1,6-bisphosphate, and so on. You're familiar with this graph for glycolysis. You could make a reaction graph, whereas you connect two reactions by an edge in a network if they share a reactant and a product. But the more interesting part is you can actually make them into a hypergraph, wherein or a bipartite graph where you have two types of nodes. So the volume is clear, right? I just noticed a chat message. Okay, so you have two types of nodes, which include your circles, which are your metabolites, and rectangles, which correspond to reactions. And glucose and ATP will sort of connect to the rectangle node, and out come G6P and ADP. ADP is not shown here, but this is closer to the representation you would have in a, say, a biochemistry textbook. This representation has a number of advantages and also a number of challenges. So the first thing is, you will not, representation is good enough to eliminate a two-step glycolysis, right? You won't be, from here you may be tempted to conclude that glucose can be converted to ADP and ADP can be converted to pyruvate, right? Whereas here, that is, you can choose not to infer that because you know, you will have multiple metabolites that need to be produced and consumed here, and you can, that information is captured here better. So we built an algorithm to study these kinds of networks. So these kinds of bipartite graphs, especially for microbial communities, and start predicting how a metabolite in one organism could lead to the production of a metabolite in another organism. So this, you can imagine, has consequences for how two organisms could complement or cooperate with each other. So what does our algorithm do? It's based on dynamic programming. So most computer science people would know what dynamic programming is and what breadth first search is, but these are just, you know, if you don't know, just keep them aside as reasonably simple algorithms. But then we had to tweak them a little bit to account for the availability of different molecules and so on. And also the fact that your metabolic networks are highly cyclic and so on. Our algorithm is available from the Python package index. What does our algorithm do? It requires only the topology of the reaction network. It doesn't require stoichiometry or atom-atom mapping. And it is quite scalable to very large networks. And it can efficiently handle cyclic and branch pathways. It also looks at multiple alternate routes of conversion that are available for any given pair of molecules. So how do we extend this, take this algorithm and extend it to the gut microbiome? The question we asked was, how does one organism support another? And we looked at one major way by which this happens. This is in terms of relieving the reactions that are sort of stuck. So there is an organism in its environment, it is able to produce a bunch of metabolites and it is not able to produce a bunch of metabolites. Meaning there are some reactions that are active in that environment and there are some reactions that are inactive because certain input metabolites for that, so those reactions are not available. But in the presence of a second organism that can give it some of these nutrients, it will be able to produce these metabolites as well. So we came up with this index called Metabolic Support Index, which basically measures the fraction of reactions in an organism's metabolism that are originally stuck when the organism resides alone by itself, but relieved when present in a community. I'll give you a quick example. If all the reactions remain stuck, there is no metabolic benefit of being in a community. And this index essentially tries to quantitate the extent of benefit that an organism receives from another. It's an asymmetric index, as you can see from this simple illustration even. But as a quick example, let us consider these two organisms that when they are alone, organism one has two stuck reactions like R4 and R6, whereas this organism has 5 stuck reactions. When in a community, through the exchange of certain metabolites, it is possible to relieve this blockage of these reactions and produce furthermore metabolites, which will probably help the organism to grow and so on. And therefore, we compute the MSI in this fashion. And here we see that all the reactions in A are now producible. So there were 2 reactions that were stuck. They are both now, they can fire, so to speak. Whereas here, there are still 3 stuck reactions. So 2 out of 5 reactions are blocked, wherein you get this index of 0.4. So what is the use of this index? We found that in experimentally observed co-cultures, whenever we found that one organism had a higher support than the other, it turned out that this organism had a higher biomass, and this basically helps us capture the sort of potential for two organisms to be in a community and to see which organism may dominate the other and so on. And when we applied it to this network of 21 organisms mentioned, we looked at every pairwise combination of these 21 organisms and constructed a microbial association network based on the MSI values. And we also looked at the exchanges of amino acids in this network. And here you see a particular part of the network which shows various interactions, and it shows that there are certain organisms that receive a lot of help from the other organisms, but there are certain organisms that give out a lot of help, and there are certain organisms that have a very interesting they are key players in the community based on the interactions, right? So we found a bunch of organisms that are quite important in the recovery of the gut microbiome. Basically the exchanges between these organisms seem to predominate in these interactions and enable this recovery. But there are some limitations to this approach. It's finally only as a sort of static snapshot of what happens in the gut. The community is ever changing, and but given a bunch of organisms, this does tell you their potential for cooperation and so on. And while some of our predictions agree with previous experiments, a lot of the predictions still remain to be validated. Right, and there are experiments that need to be done, and we can also look at literature for validation and so on. The algorithm itself obviously depends upon the quality of the input network, and that's always a challenge. And we still don't weight or rank these metabolites and paths. That's something that we could do, which could improve the reliability of some of our predictions. And we also don't identify very long pathways, but that's probably not so important. The last limitation, of course, is that we look at this whole thing through the lens of metabolism. And while I've convinced you already that it's important, it's certainly not the only thing that's happening. There's a lot more that's happening. So to summarize, we built a tool that identifies metabolic exchanges and biosynthetic pathways across organisms to try and identify the metabolic crosstalks in a community. And we came up with an index that is able to quantify the support that one organism receives from another. And it's a generic framework for understanding any microbiome or any community. And this can be helpful in, for example, studying microbial consortia or identifying different microbiomes such as the gut or the skin. Or, if you've seen this recent initiative called MetaSub, which talks about, which basically identifies different, the microbiome load in a subway, right? It's actually happening in Hyderabad currently, and we are trying to get it started in Chennai as well. And this has already been in the New York subway to find the microbiomes that are present in different, the metagenomes that are present in different parts of a particular rail network or train network. So there are lots of people and collaborators who've helped in this work. So Reaction Miner, the main work was done by Arvind Shankar, a very smart dual degree student from computer science, and Dr. Shayan Ranu, who's now moved to IIT Delhi. NetGenes and the prediction of essentiality was done by Karthik, who's now in Germany, and Ravindran, who heads the Robert Bosch Center, who's one of our top ML-RL guys. MetQuest and the microbial communities analysis was done by Aarti, and inputs for the algorithm are given by Meghna from computer science, and the experimental data on which all our predictions were done was supplied by Niranjan Nagarajan. And for more info, you can reach out to me on by email, or you can look at our lab website, our GitHub website. And these are some of the papers that discuss the things that I presented to you today. And do check out the NetGenes database as well. I do see a few questions on the database as well. And that's it. Thank you for your attention, and I look forward to taking questions from you.
Dr. Dr. Karthik for the very interesting talk. So would that be okay if we take the questions towards the end, or we could take questions? We could take questions for this session here, and then take the questions for or we could clump them together towards the end.
Either way is fine. I think it's.
I think you can finish it so that yeah. So
Okay.
some questions Karthik can answer
Sure.
now.
Sure. So okay, so then we'll just move on to the questions. So first question is from Abhishek, and he's asking: essentiality of a gene is usually dependent upon changing external environment. How does a static network analysis take this into account?
True. So we basically use a particular definition for essential genes. So we basically predict essentiality in a rich medium, right? So it's, I mean, it's obviously quite contingent on the environment and things like that. But for the purposes of this study, we had a particular definition of essentiality. So, which is sort of standard across the many of the studies that try to predict essentiality. Of course, right? Even in the it's highly contingent on the environment, the presence of different nutrients and things like that. So, this is a broad picture of essentiality. Yeah.
Okay, so the next question is asked by. Sorry.
Oh wait, I have another question that was on private chat. So, for centrality-based essential nodes identification, is it okay to consider network known? Is it okay to consider a network of known DG expressed genes or complete interactome? I think the complete interactome makes it better because I mean more useful data for the network to learn from. Right, but it's also possible to train it on some of these subnetworks, although we haven't tried it. But the complete interactome is seems to work reasonably well. I think we have fairly good accuracies, more than I think 85%, if I remember right. And there is a question on, I don't know, can this algorithm help to study learning disability? So I'm not sure which of the algorithms it is. Maybe they could clarify. And there's another question on can we use this to identify interactions among different communities, say gut-lung axis or gut-brain axis? Yes, very much. So we can, I think it's important to, in some of those cases, look for the metabolic signatures of those diseases as well, right? So what is the metabolic disturbance that's happening in that disease? And what are anyway we look at metabolism, right? So all of our analysis is centered on metabolism. So can we actually look at the metabolic signatures of that disease and then see what is the connection between, you can in fact model it. There are other tools also that can help you model the host pathogen microbiome complete system, right? Using similar tools that I've talked about and also say constraint-based modeling. So all these things are quite possible. And I think I missed another question. Since LP allows a wide range of constraints, can LP help with the limitation of dynamic gut environment? It is possible though. I mean, if you see, a lot of our work happens on constraint-based modeling. And I, only recently I've started speaking a little more about graph-based modeling, simply because we do not have accurate enough networks that can be thrown to the constraint-based models. So that is why
Dr. Karthik, sorry, do you mind if we read out the questions completely so that everyone else also knows what the question is? So would you mind if I just read it out?
Basically, use of this was on the entire chat, but yeah, I think I can still repeat because
I can do that for you so that you can just so that you don't have to keep browsing through the chat. So I could do that. I could just read out the question for you. Okay, so Dr. Gayan asks, is the software available freely and what is the computational load for the software?
Yeah, the software is available freely. It's open source. You can check it out on both the Python package and on GitHub. And computational load, I think it works very quickly if you're looking at pairwise interactions. You can also look at larger communities and networks, but there it can be challenging because if you may want to expand. We've reported for quite large communities, and it takes it's less than a few minutes even for a large community. But then if you typically want to look at a microbiome and you want to pick, say, n choose 2, right? So here we did 210 pairwise combinations. So that can be quite computationally expensive. But every single analysis doesn't take too long. But you may end up having, say, I want to simulate 500 pairs or 1,000 pairs and things like that. So then it starts adding up. But because it's all independent, you can easily parallelize it. You just put it on a cluster or something, it can happen very quickly. So it doesn't matter.
Okay, so there's one question on, can we study H. pylori infections and forming ulcers to cancer? I think this is a general question. So
yeah, so I think there are some studies that have already seen that. Maybe the kind of mechanisms that you want to uncover with that study is not readily visible through this kind of analysis. You may want to look at c-Met signaling and things like that if you want to study H. pylori infections and so on. So there are other methods that people have been used, but you can obviously see the metabolic signatures of two different scenarios, right? So maybe you have the microbiome of healthy people and you have the microbiome of H. pylori patients, and then you can sort of see what is the difference in the metabolic signature. So those things are quite possible.
Okay, so we have the last question, which says many microbes produce a lot of metabolites. Do nearby microbes use all the metabolites that are produced in the environment? Microbes will only choose what is required for growth. Is the assumption that all the metabolites secreted can be used by all the microbes?
Yeah, so there are two layers to this problem. Do the microbes have the ability to use them in the first place? Meaning, do they have a transporter, right? B, yeah, if there is a transporter and if the metabolite is available, will the microbe take it up and use it? Maybe only when it's important for growth and so on. So this becomes a classic argument between a constraint-based approach versus a graph-theoretic approach. A graph-theoretic approach doesn't so much worry about whether it's going to impact the growth and so on. But this is a very good question, right? So the idea is to use both constraint-based approaches and graph-based approaches if possible to complement each other and get more insights about a community, because the final goal is to understand what kind of interactions happen between a pair of microbes. So this is I don't have a direct answer to this. Of course, in this model, it is assumed that everything that is in the environment and can be uptaken will be uptaken, right? No matter whether it's important for growth or not.
Okay, so that I think we have no more questions for Dr. Karthik, and now we'll, 1 second. Okay, so then now we'll move on to Dr. Venkatesh, and just to briefly introduce him, Dr. Venkatesh is continuing professor at IIT Bombay, and he did his PhD from Purdue University, and he has more than 2 decades of experience in systems biology, network analysis, synthetic biology, metabolic and regulatory networks research, and currently his lab works on metabolic engineering and quantification of biological networks, including genetic signaling and metabolic pathways. And yeah, interestingly, Dr. Venkatesh, Dr. Karthik, along with Dr. Nagasuma Chandra, who is a faculty at IISc, they're also co-founders of a company called Q-Biome, which offers cellular, metabolic, and process engineering solutions for biotech, pharma, and chemical industry. So with that, I would like to request Dr. Venkatesh to please start his presentation. Thank you. Dr. Venkatesh?
So, are you able to hear?
Yeah, we can hear you.
Yeah, hello everyone. Thanks to Surat for that introduction. So I'll quickly get to something about systems biology that I want to share with you all. So Karthik talked about network analysis, both graph theoretic and constraint based. So we also do constraint based kind of analysis, but I'll be focusing more on the kinetic modeling. And then probably you may see some similar approaches in your previous web seminar that you might had with Vantage Biotech about pharmacology, systems pharmacology, right? So something similar, but I'll give you a brief about what systems biology is, and then look at some of the things that we have been doing. So as biologists, our goal is that if you know the complete link between genotype to phenotype, then we understand the system well. And then, are we able to predict, given a particular system, biological system, what phenotype that it goes to is always a challenge. And then, and then we know that presence of a genome may not ensure a phenotype, because it requires a specific state in this hierarchical chain, right from the genotype to the phenotype. So I give this example of, the central dogma of biology, where you have a firefly, where you have a phenotype, a metabolic reaction should take place. Because it does not happen thermodynamically under ambient conditions, you need a catalyst, which is a protein, then you have a RNA and you have a gene, right? So you need to have this connection between luciferase gene getting decoded to mRNA, then you have to catalyze the reaction has to happen, and then you'll have the firefly glowing. So in 1983, this was transferred to a tobacco plant, where a luciferase gene was activated in tobacco plant, and then its gene was decoded, the enzyme was kept stable, the reaction happened, and then you see that the phenotype of the tobacco plant glowing, right? So this was done in 1983, and there was no computation involved. It was purely by trial and error that a phenotype of one organism was pushed into a phenotype to the other organism. Okay, so here a lot of trial and error experiments were done before they were successful to demonstrate this transfer of phenotype from one species to the other. Okay, so if you look at historically, right, so biology has always been a descriptional science. So you keep on describing, because you don't know the system, you want to understand the system. So you break open the system and look at all the components that are present. And the last 4 or 5 decades, molecular biology has given molecular level description of this hierarchical state that we have been talking about. And that results in genetic signaling and metabolic networks, like Karthik was indicating. So you have a lot of interactions between different components. And this is the result of the reductionist approach of molecular biology. So because we are reducing the system and looking at all the components that are present. And bioinformatics, through the pattern matching and other approaches, adds more to this information. So this results in a highly interconnected network. And so we want to make sense of this interconnected network. So there are different approaches that one can use, right? So one could be graph theory, Boolean analysis, it could be constraint-based analysis, right? So there are different ways that one can look at these complex networks to try to make sense. So if you look at systems biology, one of the approaches in the systems biology is trying to see how these systems operate under a changing environmental condition. Okay, what are the phenotypes that it takes given a changing environmental conditions? So as an engineer, we are familiar with quantification at different levels. Okay, so if an engineer looks at a system, he has completely designed it, so he knows how to optimize it. He knows the each component, why it is placed, what is the role of each component. So he understands because he has built it. So engineering systems are quantified to the level that we design it, we optimize it, we optimally operate it. Okay. Now, obviously, in the last 2 decades, biology has been evolving to be a highly quantitative science. The omics data, for example, throws open different levels of data. Okay, at both time and space, you can get a lot of data now, and which is quantitative in nature. And now is the time that you can apply system science, which engineers and physicists are familiar with, to these systems, and that is what is systems biology. So if you want to look at how the data makes sense in terms of the known design of the system or the known components of the system. So if you think in terms of an engineer's viewpoint, you essentially design a system, then you operate the system, then you would want to control the system, and then you would find some faults. And then if there are faults, you go and redesign, and then you evolve to a new system. Right? So any engineered system you look around, it may be car, aeroplanes, satellites, computers. So have gone through this cycle of designing, operating, and then you control up to specific conditions, and then you may find some faults. Then you go back and redesign, and then evolve. Right? Biological systems also go through these stages, but obviously in the evolutionary scales and driven by evolutionary dynamics. Okay, so that's what drives the evolution of new systems to adapt to the changes in the environment, or to evolve to take care of perturbations in the environment. Okay, so they also go through a similar phase. So if I look at an engineered system, which is a bottom-up design of a complex system, right? For example, an aircraft. So it has about 1250 computers. There are hundreds of feedback loops. you have millions of components present, right? But because we have designed it, because it's a bottom-up design, so the design manual is available for us. So right from the Wright brothers, okay, who started the design of this aircraft to the modern aircraft, we know how it has evolved because humans have designed it. So each change or each component that you add, you add with a purpose. And that is the reason why a design manual exists, right? So now it pretty much, only takeoff requires manual effort, right? So it is able to take care of a lot of sensors on the, at different places on the aircraft, which measures the pressure on the top, pressure on the bottom. The hydrodynamics is calculated by the computers. It tells you where, how to keep the aircraft stable. It will tell the pilot, when a turbulence can come, and then he can warn that a turbulence might occur in the, in the near future. So these kinds of feedbacks are fed into, but there are a lot of calculations that are happening, right? So there are, because of all these feedbacks and the calculations that are happening in the computer. So there's a lot of calculation each instant that is occurring to the perturbations in the environment to keep this, system stable. So what is the phenotype of the system? So you essentially have to take off, keep it stable in the air, and then you have to make it land, correct? So that is the essential property that you want. And obviously you have weight, that it has to balance a certain amount of weight that you have to transport from one place to the other. Right? So that is the phenotype of this. And then we know how this system operates. Right? So you look at E. coli, right? It's one of the well-known system that is quite well understood. Right? So this is a design in nature. So there's about 4,400 genes. There's a lot of connectivity between genes, mRNA, proteins, and metabolites. So there are thousands of feedback loops, right? So the modern aircraft had hundreds of feedback loops. So this has got thousands of feedback loops. And there's no design principle available because we have not designed it. So in nature, it already exists. So there's no manual for E. coli, right? Unlike the engineered system, you don't have a manual for this system. And there's no computation. I mean, that's what you would think that, E. coli does not have computers, right? The aircraft had a lot of computers to calculate each one. Right? So you think that, how can E. coli sort of compute? So there's no inherent computation, as is what you would think at, or external computation that you would think of, right? But there are a lot of control, sensing, and interactions that are taking place because of all these feedbacks and connectivity that you have, okay? The complex connectivities that you have. So E. coli appears to be more complex than the modern aircraft, purely in terms of the components and the interactions and the feedbacks that are present, okay? So it is far more complex. And the aircraft had a defined phenotype, right? It had to take off, be stable, land, and transport, some people and load from one place to the other. But E. coli has lot more phenotype to demonstrate. So it depends on the environmental condition that it is in. So if I put it in a reactor in a very controlled atmosphere, it will show one phenotype. I put it with Bacillus, another strain, it will have its own phenotype. So it can show multiple phenotypes to survive and multiply in the environment which is constantly changing. Okay. Say, for example, you might have heard of chemotaxis, right? So chemotaxis is essentially E. coli moves from a lower concentration of glucose, which is its food, to higher concentration of glucose. Okay. So how is it able to do this, right? So if E. coli is able to do this, that means that there has to be a design inside. Okay. So this is the fundamental, definition of a what is called as a cybernetic law. So the definition of a cybernetic law is if a system is able to show a property, then the design of the property has to be inside. Okay, inside the system itself. Okay, so if I'm able to see, that means that the design for the capture of the image, right, has to be inside me. So I need to have a retina, I need to have a camera, I need to have focusing. So all those have to happen inside me, right? So similarly, if E. coli is able to do this, then it has to have motion because it has to move towards it. So it has a motor, it has a flagella, there is a sensors for glucose. It measures glucose at one point, it moves randomly, measures glucose at another point, takes the difference, remembers the old glucose concentration, shows that it is a positive gradient, and then moves towards it, right? So it's a very engineering thing to do. If you look at it, it's like, an engineer can design chemotaxis where he has sensors, it measures the sensors and then randomly moves, again measures, compare the concentration before, and then sees whether, it has to move in that direction or not. So E. coli already has it. So now we understand each component that is there in the chemotaxis of E. coli, how it does it, right? What is the time constants for the signal to go from the sensor to the motor, how the motor operates, what are the clutches in the motor, okay? What are the energy ATP molecules or the H+ molecules that are needed to make the operator work, correct? So now we can build a manual. So I can now start writing, right? So if this E. coli demonstrates chemotaxis, so what is the subnetwork and the interactions that are taking place inside the system that gives it this particular phenotype, okay, of the cell moving from one point to the other. So this is what is systems biology, where we're trying to get the design principles for each of those phenotypes. So there are a lot of students here, so I just give this typical example to illustrate, what is the complexity or what type of systems that we are looking at. Okay, so for example, if I take a red ball and a blue ball, and I randomly pick, a blue ball, right? So what is the probability that it is a blue ball? Right? So obviously it is 50%. So there's a 50% chance that it is a blue ball. Okay. So if supposing I change the experiment a little, so what do? I pick a randomly a ball, and in the next step, I put the same color of the ball that I picked in the previous step. Okay. So if I do that, then I will have two blue balls in the next step. If I pick a blue ball, next time I'm going to have two blue balls and one red ball. Now, after 50,000 step, I want to know, what is the probability that it is a blue ball. Okay. So I hope you understand that, I start with two balls, but then I'm going on increasing with, higher number of balls. Oops. I'm sorry about this. I don't know, something happened. Yeah. Okay. So to cut the story short, so I can always bias towards blue, right? So if once I pick a blue, I can always bias it towards blue. And then I'll see that the probability that, I may have 49,999 blue balls and one red ball. So then the probability that you're picking a blue ball is almost, close to 1. But by chance, I could have also biased it towards red, right? So I could have had 49,999 red balls and one blue ball, right? So that also is a possibility. Then I would have gone to almost, zero probability that it is a blue ball, right? So what has been done? So I start with a 50-50 chance, but then I can go to any probability, right? I can go to any probability depending on the history. Okay, the past steps can drive me to almost close to 1, may drive me to almost close to 0. If it is alternate blue, red, blue, red, then I can stick at 50, right? I can have any probability. So this is, I have introduced a nonlinear step by picking a result, a outcome of the previous step into the next decision making. So I've introduced a nonlinear step. So when I introduce a nonlinear step, I can have multiple steady state. Hello? You're able to hear, right?
I can hear you, Doctor.
Yeah, okay. So I can go to multiple steady states, right? So I start with the same state, but now I can go to multiple steady state, and it depends on the history. So I can have multiple outcomes possible depending on the history of the steps that I've taken. So this is the property of a complex system. So any complexity can go to multiple states. So we all can start with, when we all start as a single cell. So I hope there are no foreigners in this audience. If I assume that everybody is Indian and from Indian subcontinent, okay. So our genes will all be very, very close. So the probability that we are all different as a system at t is equal to 0 in our life, okay, will be very, very small. It will be 0.001. The SNPs that we carry from our forefathers is what will be the difference. Okay, other than that, the system will not be very different. Okay, we start almost with a similar state, but we all end up with different phenotypes. Okay, somebody is tall, somebody is fat, somebody has different metabolic rates, right? So we have all sorts of variations that we come to based on both the effect of environment and also the effect of the system itself, the variations in the system itself that can drive us to different steady states. Okay, so this is a fundamental property of complexity, and then this we have to appreciate when we are trying to look at biological systems. So the properties of complexity is that you have multiple steady states. It depends on history, as I said. So that you have got parametric, and they may be sensitive, and the sensitive may be different in different individuals. Okay, you have something called as bifurcation. So you keep changing, and suddenly it shifts you to a new steady state. Okay, you can have systems which are robust. You vary a lot, but even then, nothing happens. You adapt, and you can come up with new designs with rearrangements to evolve to a new system, right? So all this is happening because of the complexity that exists in the system. Okay, so you want to build a manual for a biological system, then you need to capture all these kinds of properties and then show for a particular phenotype or multiple levels of phenotype what is the actual design? Which components are coming together? What is the time constant? Okay, what is the binding constant? What are those concentrations of protein? So you can imagine the complexity that you are looking at to, to understand the system as a whole, to connect all the genotype to the phenotype and the linkages of the external perturbations that may be happening. Okay. So engineers and physicists have always been looking at nonlinear dynamics, multiple feedback loops, multiple interactions, cascade structures, feed-forward loops. There are interactions between modules, right? So there are two modules in different, disconnected by space, but they're talking to each other, right? You have time scale separations. Something is happening in seconds, something is happening in minutes, something is happening in, years, months, right? So you have different time scale separation. So there is a flower that blooms in Kerala, which blooms once in 12 years. Okay, so every 12 years, it blooms. Okay, I forget the name of that flower now, right? And between March and April, so the error is about 2 weeks time. Right? So how is the system able to do this, right? So it should have a clock to keep track of that 12-year, cycle. Okay, so that means that the design has to exist inside. So there is some time scale separation happening inside a design that keep tracks of this time, and then all the hormone release and then the flowering and all that happens precisely in that time scale, right? So this is what I mean by saying that building a manual, trying to understand what is the design that exists inside the system which captures that particular phenotype that we see in a biological system. Okay, so what we have to deal here with is biochemical networks and the sub-networks and the motifs that give rise to different structures and dynamics. Okay, you can look at different motifs, interactions that may be happening. On the left side, I've given you different motifs that exist in biological systems, and they can give rise to, sensitive response, amplification. It can give bistability. It can give oscillations, adaptive systems, homeostasis, right? So all this input-output responses it can give based on which motifs operate under a certain conditions. How noise has to be handled. So nature has been evolving all this to take care of the redesigning of the system to take care of a particular environmental perturbations that might be occurring. Okay, so we want to now have to do a map in terms of a phenotype to a structural, to a dynamics, to a structure, to a motive. So we need to bring these linkages so that we go right to the components that are present in a biological system to a particular phenotype, the dynamic response that the organism yields. Okay, so this is the role of systems biology, that we start building manuals to say how a particular performance or a output is being shown, and what are the design that exist inside, okay, which are evolved naturally to take care of that perturbation or that particular phenotype. Okay, so I can show many of these properties by just rearranging some of these components, and like in many things in biological systems, there is a lot of diversity, right? So for the same adaptive response, you can have many designs that have evolved in different systems. Okay, so all this comes about because of interactions, transcriptional control, signaling network, metabolic changes, mutations that occur, allosteric interactions, conformational changes. So all this that are occurring at the molecular level, sort of give rise to final input output that you see. Okay, so what is the first step? So okay, now we understand that these systems are complex. We need to know the components first. So that is why systems biology could not have come in 1950s. It's now gaining importance because now we understand some of the components of the system. So the molecular biology has broken up and then given us the components and how they interact, right? And now omics data is giving you all the quantification. So that is now I'm ready to apply system level kind of approaches, right? So before that I could not have done it. Right? So the first thing that we need to do is reconstruct. Okay, so all the cellular functions I need to reconstruct. Then I can look at what is the role of a component. I can apply modeling and simulation. I can do feedback analysis, and I can now start linking the system to the particular phenotype, incorporating the effects of the perturbations in the environment. Okay, so that's the first step. So I need to reconstruct and then build and then start looking at how the analysis. So for this particular talk, I will talk to you about in human metabolism, and then how we can model those and to capture health and disease. Okay, and we apply these principles of systems biology and control analysis to find out, how can we characterize, human systems. Okay. So if you want to capture the cellular processes or phenomena, physiological processes, then systems biology, as I said, once you bring all the components together, there's a lot of mathematical modeling and computational analysis that you need to do. You need to do parametric estimation. So all those system level analysis that one does, we need to capture those. Okay, so we have to apply a lot of computational tools. And the idea is we want to do a quantitative mechanistic based model. So there's another way of analyzing these input output, right? So, and which is in the, in the news nowadays, that is the artificial intelligence and machine learning, right? So I can take a lot of input data, I can take a lot of output data, and I will just say, what could be the, what is the correlations of this input output, right? I need not know any mechanistic details, but I can still get correlations between input and output, saying that if these four inputs are high and these four inputs are low, then something may happen, right? So I don't care about a lot of mechanistic details. So that is one way of looking at it, but we want to capture the physics behind it. So we want to look at the mechanistic. I want to capture as much mechanisms that is there, that is known, and which are relevant to a particular phenotype to be captured. Okay. For example, if a transcriptional factor dimerizes before binding to a DNA site, I want to capture that dimerization because it may play a role in the particular phenotype. Right? So that's what I mean by saying that I need to capture the mechanistic detail. Okay. So because only then the biology of the why of the biology comes out. Okay, because you have captured the mechanism. If I do pure AIML kind of input output analysis and then build a black box model, then you will not know the mechanistic insight, but you will get insights about how the input output are correlated. Okay. So so we want to start with molecule, go to pathways and go to cell, different cell types causing tissues, and then finally to the phenotype, right? So you start in time scales, different time scales you want to capture, and different time scales, spatial scales also. So both temporal, okay, and time scales you need to, spatial and temporal changes you need to capture. Okay, so right from the molecules to the whole physiology is what you want to capture this diversity that exists. So that is the challenge. That's a challenge, and then there is a a beginning that has been made at different levels. For example, you have liver models, skin models that have started coming, accounting for different tissue types and the physiology that you want to capture. Okay. So what is that? If I'm able to capture the physiology, one of the ways that I want to analyze is obviously the disease state, so that I can either manage the disease or come up with some cure of the disease. So in such a case, right, so you have a complex system with all these interactions where you have environmental perturbations, diet, lifestyle, all causing some steady state that it can go to. And then, you have the system at different levels. It could be at a reaction level where metabolic reactions are taking place. It could be communication where the signaling proteins are taking, a role. And then you have interaction, where different transcriptional factors may be interacting, okay, at the genote level. So the disease may happen because of a perturbation at any of these levels. It can start with a singular problem, but it can go to a new state where a metabolic disorder can lead to a signaling, and then some mutations can be selected, and it can suddenly become a genetic perturbation as well. Because these are all dynamically readjusting design systems, which readjusting designs. Right, so the problem may start somewhere, but it can start showing up at different levels. Okay, at signaling, it may start in signaling, but metabolic perturbations can happen. Can happen because of that. Okay? So that is what we want to try to see whether we are able to get these connections. So the approach that we have been taking is we do a lot of data mining, build the networks. Okay, try to capture all the omics data that may be available. Right, and then once we have protein-protein interaction, signaling network interaction, transcriptional networks, metabolic networks, so we see which are the key components we need to account for a particular physiological state, and then we do the modeling. We build all the mathematical models, and then see what kind of models. So some components can be at steady state because we don't know kinetic information. Some other system may be dynamic, and then we so this is quite challenging. That how can you build a model given this complexity that exists, and the type of information that you want to include, and some of the information that you don't want to include, right? Because you have a particular purpose in building a model. Right? So you start with data mining, collect a lot of information, build networks, then you start writing mathematical equations to build the model, and then you link it with the experiment to get a parameter, and then try to validate some of these clinical trials. And then if it does not match to some, then you go back and then look at whether we have missed something in our data mining, or you can come up with hypothesis. And then this can lead to personalized medicine as well, right? So if you are able to link everything and then get all the key components that may be present, so you can hypothesize that down the line, if you're building such a platform, so you can try to link clinical data of an individual or at a population and then try to rationalize it. Okay, so that's the approach that we have been taking. So in the last 20 years, we have looked at models of liver, muscle, adipose tissue, skin, GI tract, pancreas, and we wanted to incorporate the effect of lifestyle. So we do have through GI tract food absorption. Supposing you have high carbohydrate, protein, macro and macronutrients, what is the effect of exercise in terms of hormonal change? What could be an effect of drug on metabolism and signaling? So we can now start looking at transcriptional factor, different signaling molecules, hormonal regulations that may be happening, so that we get a systemic understanding of disease. We can predict role of diet, drug, and lifestyle in any disease progression. We can engineer drug and then manage diseases. And we can personalize, and not only that, we can look at a population level. Okay, so most of the drugs that these pharma companies release, in many instances, is only 25-30% of the people it works. So they know that it will not work in many individuals, right, at a population level when you look at it, right? But still it has to be released because it's going to benefit certain number of people. So by trial and error, you need to now identify on which individual this particular drug, the efficacy will be high. Okay, so can we rationalize that thought process? Okay, so then such models will be helpful in trying to find out which subpopulation a particular drug may be helpful, or with subpopulation some drug may be toxic, right? So these kinds of questions we can start answering if you have a platform of this kind. Okay, so we look at I'm a chemical engineer, so if I look at a chemical plant, I have a lot of processes, right? A reactor is there, a separator is there, a purifier is there, distillation columns. So we all have a network for these components. So similarly, you can write a network diagram, right? So for example, insulin signaling, glucagon signaling, how calcium signaling is taking place. So this is essentially insulin glucagon network that I'm talking about, and they all go and talk to the metabolism, right? So it is controlling the metabolism. So I can go and look at now metabolic network, how glycolysis is taking place, what is the urea cycle, TCA cycle, cholesterol metabolism, energy metabolism. So I can now try to see what is the state that this metabolism is given a particular perturbation, right? And then link these two and then get a sort of a holistic picture or a system level picture of how a particular state is going, right? For example, liver cells, hepatocytes, what is the state they are in under a particular condition, right? If supposing alcohol, right? So there's a perturbation of alcohol, somebody drinks alcohol, then the hepatocells, what perturbations they go to, right? So we can start asking these kinds of questions, once we have a platform, right? So I can look at, somebody has exercised, has taken 50 grams, I've written it in terms of glucose. So somebody takes 50 grams of glucose in breakfast, lunch 100 grams, and he again exercises in the evening and he has dinner. So then I can start going and looking at these dynamics, how glucose goes up and comes down, how insulin plays a role, how glucagon, what happens in muscle cells, what happens in liver cells. I can look at different spatial levels and then time and then try to see what are these dynamics that are happening. So these are plasma levels, right? So in blood, how things are changing. So now I can start and correlate to actual clinical data. So I can go take clinical data and then map onto this. Right? So these kinds of things are now possible. So as engineers, one of the first things that you can do is look at input-output correlations. This is at steady state. So this is a steady state analysis. So for example, in 10th standard, you might have done an experiment to see how current flows with respect to voltage. Like voltage is the input, okay, that is the potential, and the flow of electrons is the output. Right? So how does that behave? Ohm's law, it is a linear curve. Right? So here I'm looking at what is the rate at which, instead of electrons, I'm looking at what is the rate of glucose entering into cells. Right? It could be muscle cells, it could be liver cells. So I'm trying to find out, quantify what is the rate at which glucose is crossing the membrane of a particular cell type and then entering inside. Okay, and what is the potential? The potential is the local environment. What is the insulin in that local environment? Right? So that becomes the potential. So if as soon as glucose goes up, your insulin is pumped in, and then about 0.1 nanomoles, right? So it gets completely switched on. And this becomes my input output. So just like the Ohm's law linear curve, right? So now you have a bistable curve like this. So this is the response of glucose being absorbed with respect to insulin. So you'll see that this is a nonlinear behavior. So switching on and switching off are two distinct levels. Okay. So this is a similar input-output curve, right? For our, to take for the glucose to go inside a human cell with respect to insulin in the blood. Okay. So what happens if I perturb a particular signaling molecule? So this is PTPase. It's one of the key signaling molecule in the insulin signaling pathway. If I double this concentration, what happens is your input, this curve goes to the right, right? So your body is producing the normal amount of insulin, but now suddenly it is not working, right? The glucose uptake is not happening at all. So the response is not occurring at the physiological levels of insulin. Right? So this is type 2 diabetic state. It is nothing wrong with your insulin. Your insulin is fine, but the signaling pathway has got disturbed. Okay, and your input output has got shifted to the right. Okay, so this is something like if supposing if the lab attendant in your school does not clean the wire or the the resisting wire, the conductor. If it doesn't clean, right, every year experiments are going on, so it gets oxidized, and then the oxidized particles keep collecting on the wire, and you keep the voltage. Now the system is different, and the slope of the curve will change. The resistance is different, okay, because the input output has got altered because of a perturbation, because something is getting coated on the top of your metal, right? So your input output will change now. So the slope of the curve will decrease because the resistance has gone down. Resistance has gone up, right? So something similar, right? So this is like an input output now I've got. I'm saying that, I have a type 2 diabetic state because some signaling molecule. So there are at least 22 such molecules which are sensitive, which can cause this perturbation. Right? So now in an individual, I don't know whether it is because of PTPase, whether because it is GTPase, it is because of PK3, which molecule has perturbed the input output. We don't know. Right? So that's why we treat them either by giving insulin or we treat them by making absorption of GLUT4 more difficult. More easier. Okay, you replace the insulin by some other drug which activates your transporters, and so that it goes in. So you either use something like metformin, a drug, or you use insulin, a hormone, right? To make it functional. So then what you have done, you have rationalized your input output to be at this state, okay, at a disease state. Actual treatment would have been that I have to bring the input output back to the original state. So if I brought this back here, then that is the right way of treating. But now in individuals we don't know what is the problem. That's why we give symptomatic kind of treatments, right? So we control either the output or we control the input. Right? The body also, once it goes into the state, body also starts producing more insulin because it sees that there is no glucose. Your pancreas is signaled to produce more and more insulin. So in the diabetic or pre-diabetic individuals, you will find higher insulin concentrations because it is trying to cope up for non-responsive insulin. Okay, so these kinds of insights now you can start getting. So one is the steady state response. The other is obviously the dynamic response. So you can actually go and look at dynamics and then look at, for example, if your oxygen supply is reduced in a cell type, what is going to happen? What happens to NADH? What happens to the ATP energy states? Okay, so this is something similar to, right? For example, you have hypoxia, right? Or you have ischemia. There's a reduction in blood flow, right? So, and in Warburg, see, you look at cancer phenotype, right? So it is so stable, right? So the healthy cell and the the disease cell, there is no difference. So that is why metabolically it is very difficult to differentiate in a cancer phenotype, the perturbations. Whereas you see in all the other cases, the perturbation is so high that either inflammation or the body gets a signal that something is wrong. Right? Whereas in cancer, this escapes. Both the immune and the metabolic and the signaling, everything, the body escapes those signals, and it goes to a steady state where the cell behave, the body or the individual or the system just adapts to that phenotype locally, and then you don't get any signals that something has gone wrong. So that's what is a sort of a cancer phenotype, where the perturbations are sort of controlled, right? In terms of the perturbation. So you can get both dynamic and steady state input output. So I don't know how much time I have. So if somebody can indicate to me.
How many how much
just some 3-4 another 5-10 minutes is what I may take.
That should be okay.
Is that okay?
Yeah, that's okay.
Yeah. Okay, so this is from our model. Another insight that came is about so if I look at the signaling pathway that I was talking about, you look at here. So you will see that you have insulin and glucagon, but you also have inputs from glucose, amino acids, and fat, perturbing the signaling pathway. Right? So I can get a input-output curve with respect to glucose, and then capture the Y as whether the system is catabolic or anabolic. Okay, you might have read Metabolism, that this is catabolic is breaking the macromolecules, anabolic is preparing or creating macromolecules. Okay, so catabolism, you start breaking to get energy. Anabolism is you have excess, you don't want energy, so you store. Right, so that is the base. So the catabolism gets activated because of pKa is the signaling molecules. Then that gets phosphorylated, and essentially the signal that is going is that it is catabolic. AKT, one of the molecules, gets phosphorylated, then it becomes anabolic. Right? So your fasting glucose level is somewhere about 5.2 millimole per liter. Okay, that is 80 mg per deciliter. Right, so that is what is your fasting glucose level, and you are mildly catabolic. Right, so if you're fasting or you're sleeping in a long duration, 12 hours, you have not eaten anything. So your body, your brain, your muscle, your heart, cells, all this needs energy, right? So your glycogen is broken down first, and if glycogen stores are over, then fat starts breaking, right, to get the energy so that it can give to your brain, and keep them in normal functioning, right? So this is what is called as a basal metabolic rate. So basal metabolic rate is, you have got enough energy to maintain all your system, but you're in complete rest. Right? So that is what is, the mildly catabolic state. That basal metabolic rate would be at around your fasting glucose level, and then it will be somewhere around close to 0.55 to 0.6. Okay, so that is where your catabolic to anabolic ratio will be. Now you get up and you start exercising. Okay, what is going to happen? Your muscles now require a huge amount of glucose. What will happen to the glucose level in the blood? It's going to drop. Okay, so if it can drop, very high, it depends on the aerobic exercise or the intensity of exercise that you do. So this will drop now. Okay, so if you're not able to instantaneously give the glucose, then suddenly you will faint because the brain will not get the glucose, the heart will not get glucose, right? So then you have a problem. So your catabolism has to be efficient. So when you start exercising, right, Usain Bolt, and he just starts running. He wants a burst of energy, and his glucose is all used up. But it has to be supplemented back because your other organs have to function. So your body becomes catabolic. So you go here. Okay, you become completely catabolic under such conditions. Your glucose levels will still be, it can even go higher, right? Your glucose level, if you measure under those conditions, it may go to 6 millimole per liter, right? But your body physiology is catabolic. Okay, because your glucose is also getting broken down and then getting pushed into the system. So it's a highly dynamic thing, right? So it is in that instant that you want this process to take place. Okay, you want that process to take place, right? So your body is completely catabolic and energy is released. So your muscles, cells, and your liver, has to become now completely catabolic. Okay, but your brain cells and muscle cells have to still be anabolic because they have to take the it will be catabolic in the sense of the energy that it wants to use. So these are all, what I want to indicate is they're all spatial. So different things may be happening inside the body itself, right? Although overall I'm saying that it is in terms of plasma levels, it is catabolic. Your body is overall in the catabolic state. So once you have done when you have done exercise, depending on the efficiency, right? So supposing I produce huge amount of ATP because of this catabolism, but I'm not able to use that ATP. So what's going to happen? Right? So my body produces huge amount of ATP, but I'm not able to use that ATP efficiently, right? So that may also happen. If that happens, your body temperature is going to go up because the ATP that you have released, okay, is not being used up, and then it has, it gets converted to heat. So your body temperature goes up, right? So your body temperature goes up, right? That is also a problem because your enzymes, your protein can become dysfunctional. So the body temperature has to be maintained. So to maintain that, you sweat, right? So evaporative cooling, your body has to be cool. So that's why when you exercise, there is always inefficiencies of the ATP that is released, right? And then because your temperature, body temperature goes up, it has to be now controlled, right? So that is because that is the sweating that occurs. So now you want to have a cooling period. So once after your exercise, people say that, you need to cool down, right? So that is, you need to come back from this catabolic state back to your fasting state. Okay, only then consume food or increase your glucose or go to the anabolic state, right? So after yoga also they say, right, you have to do savasana, right? 15 minutes, 20 minutes, half an hour, you have to just lie down. You go back to your fasting state. You go back to your, 5.2 millimole per liter through proper balancing of the catabolism. Your signaling state, hormonal state has to come back. Okay, then you consume food, then you become anabolic. Okay, so if your glucose goes beyond a particular point, it gives a signal that it has to be completely anabolic. So this is where all the fat storage, your amino acid synthesis, muscle formation is all happening here. It's completely, the signal is now it is completely anabolic. Okay, because you have given sufficient amount of glucose, and for that it has balanced your ATP requirements. It has gone beyond the basal metabolic rate, and now it can store, right? So that's how it becomes anabolic. Now you see it takes a different route. Okay, there is a feedback here which causes this hysteresis, and then it goes to a different state and then reaches this fasting state. So for a longer period of time, once you cross this threshold, you take a longer period of time to come back, and then you are anabolic for a long period of time. Okay, so you take a longer period of time to come back to the steady state. So what is this input output? So I've given at 8 millimole per liter, it drops, right? So but this is a highly adaptable system. So some individuals, it falls right here. Some individuals it falls somewhere else. Okay, so this is a very dynamic and personalized kind of input output that happens, right? And it is adaptive also. So I can change my lifestyle and then change this input output. Okay, because those designs are there that I can change some of these input output, okay, through lifestyle, right? So if I now decide that from tomorrow I'm going to do exercise, I can change this input output curve, right? Because it adapts and goes to a new state, right? So we can get, so this is the operating curve. Now I'm talking like an engineer. Like this is the operating curve of insulin and glucagon in terms of the catabolism and anabolism, right? To relate the input and output. Now I could have had amino acids here. I could have had fat here. I could have had some other hormones, ephedrine in terms of the exercise. So I could have had any input here, but I can get a input output curve. What is it? This opens up in terms of the type of questions that you can ask. Okay, in terms of applicability, right? For example, you have seen dietitians giving you an advice saying that you need to eat small quantities in multiple times, 5 to 6 times, right? Ayurveda says you need to have only one large meal. Okay, so each dietitian or a purpose gives their own pet lifestyle advice. Okay, but we can now rationalize. It is all, everybody will be true, but it is circumstantial, right? It depends on the circumstance that they are giving advice to. If supposing this falls right here, okay, so supposing this curve were to fall right here, then the person has to eat many times, because if he eats even a little calories, he becomes highly anabolic. So then that individual has to eat multiple times and go back to its steady state and not be anabolic for most of the time. Okay, but if somebody has teenagers, for example, they have a long drop. It can even drop at 12 millimole per liter. Okay, so then you come here, go back, come here, go back. Your anabolic requirements are satisfied here, right here. Okay, you don't have to go and have a very high anabolic synthesis rate, right? So you go and come back, so you don't fall at all. So such individuals, they can be one heavy meal. That's good enough. Okay, and in terms of the area under the curve, that is the best for such individuals, right? So you can now rationalize the various lifestyle in terms of a operability of the curve of the system. Okay, in terms of an input output. Okay. So these kinds of things you can you can look at what are the state, for example, what is the harm. Yeah. So to continue, I was trying to capture the how the cells can go from a catabolic to anabolic state, and the operating curve that you get with respect to some input. I've just shown it for glucose here, but it could be with respect to amino acids. It could be with any other variable in the system that you're looking at, and then try to capture the hormonal balance that takes place in terms of catabolic and anabolic. So now it is known that this imbalance of catabolism and anabolism is what brings about oxidative stress and then many other disease phenotypes, right? So obesity can also be sort of explained with these kinds of input output operating curves. So, for example, under fasting condition, you can now actually go and get the rates at which some of these processes are taking place in the signaling pathway. Right? You will see that the inputs are glucose, amino acids, and fatty acids, which are the three core macronutrients. Right? And then you have the insulin and glucagon are the key hormones that are playing a role. Then you can actually go and show that the catabolic to anabolic ratio is about close to 51. That is at a steady state, right? slightly catabolic, and then you have the breakdown of fatty acids and amino acids and gluconeogenesis happening. Okay, in terms of metabolism. So interestingly, you can now by linking the pharmacokinetics of caffeine, for example. So if you take coffee, okay, how will this pathway change? And so now you can capture. So caffeine goes and perturbs this at the signaling level, okay, the caffeine in your coffee. And then you will suddenly see that the ratio, which was about 0.52 to 0.55 under the fasting condition, has gone to 0.8. It is almost like, you're exercising, right? So you have taken coffee, your perturbation is from a molecule, but the body responds, okay, as if. And that is the reason why it wakes you up, correct? Because now the catabolism is activated. The caffeine activates the catabolism. I can now start looking at these levels and then see what is the rate at which all this feedbacks, the rates, the crosstalk between these, how it leads to a particular homeostatic steady state, okay, of the catabolic to anabolic ratio. Right? So this is the kind of insights that you can get if you have a platform. Which captures the physiology behind it, the network dynamics behind each of the physiological state. Okay, so you can now go and look at the metabolism. For example, in liver, what is going to happen if you take caffeine and your fasting state? So you will see gluconeogenesis goes up. You're already fasting, gluconeogenesis is happening, but now 40% more gluconeogenesis is happening because you have taken coffee, right? Fat oxidation and ketone formation goes up. Okay, your cholesterol synthesis goes down, your protein breakdown goes up, amino acid formation and uptake goes up, right? And then you will see that the cholesterol is not being thrown away from the bile. That means that cholesterol is rotating inside the body. Right? So these kinds of physiological insights now you have started getting in terms of a particular perturbation, an external perturbation that you have taken, right? In the fasting state. So you get up, you don't have anything, and then you take coffee. So this is the effect that you're going to get. Okay, it's highly catabolic, right? So your catabolic ratio in that curve, if you remember, so your exercise could have taken you here, right? Your coffee has taken you to 0.7, close to 0.8, right? So your coffee has taken you up, and you have become more catabolic, right? So this is the physiological effect of caffeine. So now we can start capturing these kinds of molecular level mechanism in terms of a response to a particular extracellular active ingredient. Okay, so I can do it with nutraceuticals, I can do it with drugs, I can look at what are the side effects, what are the metabolic side effects that a drug can give. Okay, or a similarly for alcohol, I can go and see what is the dynamics, how things are changing. So these are the kinds of insights that I can obtain. Okay, so based on this, we have developed various platforms. So I have a company called MedFlux, which looks at we have converted these systems biology, not the AIML. Okay, our approach is different. So we are coming up with a web platform which looks at overall wellness and predicts disease individuals. And we have already done it for close to 2,500 Indian individuals. So for Indian, again, data is not available for Indians. So we have to actually take some of these data and then try to map with the models that we have. So that is one of the services that we think that we can give through MedFlux, where people can put their lifestyle details and then they will get a wellness score, right? They'll get their catabolic anabolic ratios, for example, right? So they'll get the physiology of their particular of their particular body by putting some inputs. Right? And then we can also use it for disease management. So we can give a advanced disease management ecosystem where we're looking at diabetes, obesity, malnutrition, stroke, PCOS, low immunity, cardiovascular disease, metabolic. How do you treat? What is the diet that you need to give to a cancer patient? So those kinds of metabolic questions that you're asking with respect to lifestyle, under a disease condition, under a particular active ingredient being taken in. So we can now start giving those inputs to healthcare givers. Additionally, because we have these platforms, we can help pharma companies in drug discovery, in toxicology, so that we can propel these kinds of novel inputs, and then they can spend less on drug discovery because they use these platforms to find out what could be the dosage, what could be the side effect, what could be the efficacy, and then which subpopulation, right? And then effect of diet. So nobody takes into account the effect of diet. Supposing I take metformin and I take caffeine, both together, right? So what side effect, what effect you will have? Does the efficacy goes down? Does the efficacy go up? Because now these are conflicting things, correct? So metformin is making your glucose go in, but now you're having caffeine, which is saying that, catabolism has to go up. Gluconeogenesis should happen, right? So supposing somebody takes a tablet with coffee, correct? What effect it will have, right? So now people, that knowledge or that awareness is not there at all in the ecosystem, right? So now we can start asking those questions, saying that, if you take an API, you take a drug, right, under a particular disease condition, what diet effect will have, what other constituents that you're consuming effect will have, right? Karthik was talking about gut microbiome, right? So can we integrate those information into our system to rationalize the input output? So it just opens up in terms of your looking at various input output characterization. Okay, and then the additional strength of this approach is it gives a physiological basis. It gives a molecular basis as to why something is happening. Right. So with that, I will I'll end and then give I'll thank you all for listening. And then if you have any questions now, I'll answer.
So,
Thank you. Yeah, I think I found, thank you, Dr. Venkatesh, for that wonderful talk. And I think I found the chat.
yes, I shared that with you.
Okay, yeah, I also got it. So maybe Dr. Venkatesh, if you could just, we could just go through the questions that were posted in the chat box, and then we could record it and send it to all the students.
Absolutely, no problem. Let me just 1 minute. I'm just going to your control.
Yeah.
I don't know why I'm not able to go.
I'll just read out the questions if that's okay. Okay, so the first question is by Smita, and she's asking, where do you get first-hand access to data to construct a primary model, for example, from insulin sensitive to insulin resistance?
Yeah, so our all the model parameters, we take, we do a lot of data mining, right? So there is a lot of data available. It may be from different sources. So at the first round, we don't care whether it is the same study. So we take from different studies and then make a map in terms of physiologically what is making sense. What is the fold increase that we see, right, in different studies. And then we rationalize and then fix a parameter to match all those physiological conditions, given a particular input, right? So it could be cell level information, it could be clinical data, it could be just physiological measurements. So whatever the source that we get, okay, we try to rationalize that our model should be able to answer those physiological understanding that exist in literature. So that is our starting point. Okay, so once we have that, we now go and actually take clinical data that is available in the public domain and rationalize it. Okay, so for example, just the insulin hormonal network quantification, right? So we have taken close to close to 80 to 90 studies, the data from there to rationalize our parameters and to get our input output correlations. Okay, so there's a lot of input data that goes in at different levels to estimate a parameter. See, finally, it has to capture the physiological understanding of a healthy state and a disease state. Okay, so till we reach that point, right? So we keep adjusting our parameters to converge. That's why it takes a lot of time to get a confidence in any of these models. Then we collaborated with a hospital, got actual Indian patient data of input output, and correlated some of the models, so that can we use this for disease management, for example.
Right. So thank you. And so the next question is asked by Abhishek, who's asking, many of these networks function at different time scales. So how to model effect of perturbations in a particular network, for example, metabolism type on other network types, for example, transcription factors.
Yeah, so this is a, I mean, the model can be built with different time constants. Okay, so this is a issue of your robustness of your solution platform, right? So depending on the time scale, right? So the shorter times you can make it go to steady state and then link to a particular network. So there are methodologies as to how to overcome stiffness in the model and time constant variations in the model. See, some of these techniques are already known in the engineering domain, right? For example, you have a model for the weather, right? So it's a huge model with a lot of parameters that you have. Okay, so some things, some perturbations happen in minutes, something may be happening in days, to show up those effects, right? So you need to, when you're looking for those effects, you make some assumptions and then converge to the new steady states or the new new states that these variables are going. So we use such techniques to rationalize the time constants.
Thank you. And there's another question which asks, which is a better way of modeling a metabolic system, constraint-based method or kinetic-based method?
Yeah. So it all depends on what is the knowledge base that you have and what approximations that you take, and then what kind of input output that you want. Okay. So for example, if you have a microorganism and you don't know any rate information, I don't know any kinetics, there's no time information given. You have taken a pseudo steady state in the exponential growth and the data is given to you. So you cannot do any kinetic analysis, right? So and under those conditions, you have to go by constraint based analysis, right? So there is no other alternative. Okay. So you need to have multiple time points to be characterized of a particular system, measured and the measured key variables, okay, also provided to you to build a rational kinetic model. Okay, so it all depends on what kind of data is available and what approximations that you can make to a given input output.
Right.
So obviously for a particular phenotype, you cannot involve all the variables, right? So if you involve all the variables, then it becomes too difficult for you to even keep track of each of those variables. So some approximations have to be made, and then that is where your experience and modeling and the experience in a physiological state comes into picture, because you use some heuristics to rationalize and then eliminate some variables, assume steady states in some variables, and then build a kinetic model, or you go by constraint based.
There is one question by Abhirupa, who asks, a lot of multi-omics data from same population might not be available. How can different data be merged, and how model from multiple data will be different from model built using multi-omics data of the same population?
So there are so this is a very interesting question, and this forms the core of the strength of it also gives you the core strength of these approaches. Okay, so I can create a population now on computer by varying the parameters. So I know that each individual have their own unique parameters. Maybe it may be genetic effect, or it may be perturbations, environmental perturbations. So whatever may the reasons, the parameters are different, but you see some input output. Okay, so I can now go and create 10,000 individuals on computer with varying input output. Okay, so I can say one individual has a very high fat lipolysis, another individual has very low gluconeogenesis, right? So I can create, because once I have a model, okay, which takes care different cell types, it takes care of the signaling parameters, it takes care of the metabolic parameters. I can now see for a particular phenotype what are the key nodes. Around those key nodes, I can create variables. Okay, parametric variability, concentration variability. I can say some individual has a slightly higher AKT concentration. Somebody has a higher hormonal concentration, glucagon concentration. So I can create individuals with different phenotypes on the computer. Okay, now I can run on the software and then do a statistical analysis which individual falls closer to which phenotype on my computer, right? And then now I can come up with subpopulation groups. So these groups will benefit, these groups will not benefit. So those kinds of analysis now I can start doing, right? So we are already offering such services to pharma companies where we do we do a population study. For example, if I have an immune model, right, with different immune strengths, I can create populations. Now, if I put an API, I can say that, okay, these are the variations that I can see. Now I can develop from the clinical data and from my simulation, okay, experiments. Okay, now I can start doing statistical analysis and find out which fall closer, which of these, fall closer to my phenotypic, outlay that I have got from my simulations. Okay, so in that sense, I can now start correlating. Okay, so that's the strength, because I cannot do 10,000 clinical study, but I can always create unique individuals on the computer with varying physiological outputs.
Right. So Abhishek also has a question on similar lines, where he's asking how much of a difference in kinetic rate constants would you expect from individual patient to another patient, and what kind of data needs to be generated to understand or capture such minute differences. This can help to use kinetic models for practical purposes.
See, for example, yeah, again, I mean, it's an extension, very interesting question, but it's an extension of the previous question. So, for example, how do you define a healthy state? Okay, so that itself is a challenge, right? So we can define diabetes, or at least we can claim to define a disease state by having some parameters or some X-ray, some ECG, some clinical parameter study. I can state that an individual has a disease, right? So a particular disease is well characterized, but a definition of healthy itself becomes you are not diseased, right? It is a negative connotation of the parameter values that you have. So I can create a set of parameter range which I define, okay, based on my modeling and sensitivity analysis of a parameter, which says that if the parameter lies between this range, okay, I can say that this individual is healthy with respect to a particular disease phenotype. For example, diabetes. I can say that if lipolysis is in this particular rate or gluconeogenesis is in a particular rate, okay, you can realize that it is not a one-by-one perturbation as well, right? So I have to perturb everything together. Okay, so individual might have a very low gluconeogenesis but very high fat degradation. Somebody else may have exact reverse or both very, very high. So it becomes statistical input-output correlations that we need to do to define what, under all things varying, what are the typical range of a parameter that I need to have to define somebody to be healthy given a particular disease state. Okay, just diabetes, for example. Okay, or just low immunity that you may be talking about. So we need to do those kinds of analysis, and that can be done now because I can create, as I indicated for the previous question, I can create 10,000 individual, okay, on my computer with various variability, and then now I say that I'm going to vary 20%. A particular parameter is between 10 to 20%, another parameter between 50 to 20%, right? And I do a study and then see how many of them go into a disease state. Right? So I can now start characterizing how sensitive a parameter is, how sensitive. I can even now generate these kinds, use these models to generate a lot of input output and then apply AI/ML on that. Okay, because I'm not doing clinical study, but I'm using computers to find out, because the advantage here is I precisely know the molecular mechanism. I know I perturb this parameter. These are the things that are going to change. Okay. Although easy to illustrate, but these are, complex simulations. Sometimes, it goes into stiff, the solution does not converge. So we need to rationalize some of these input-output or perturbations as well. Okay.
And we have a last question from Venkateshwara Rao, and who's asking for bioreactors, how to optimize productivity levels using systems biology tools. Any tools or softwares available for this?
See, the problem is even if a software is available to you, right? So you need to characterize your system properly. Okay, so I can give you a structured model for growth of a microorganism. You may be using microorganism to produce a product, and I can give you a structured model, right? Which can be used to optimize your fed batch. You can use to optimize even at the cellular level, using constraint-based models or metabolic engineering. So you can use multiple levels of optimizing, a reactor. Right? But you need to quantify your system properly. If you're able to quantify, there are ways of converting your batch system into a fed batch, and then optimizing your productivity. You can optimize the physiological state, what is the NADH levels, or the metabolic state of your organism, which yields the maximum, and you can start trying to create those situations inside your reactor so that you can optimize your fermenter runs. Okay, so it is possible. The only thing is you need to characterize your system properly. So you need to do some experiments to quantify and get the variables or the parameters for your system. Once you do that, then there are ways of engineering ways of optimizing your productivity in your reactor. And then in that way, you can be consistent also. You can avoid contamination. You can increase your efficiency of operations. You can automate your reactor runs. So all those things are possible. Yes.
I think those are all the questions we had. Thanks a lot, Dr. Venkatesh. So we'll also be sending another maybe a questionnaire to those students who probably couldn't ask the questions which they wanted to, and we could share that with you, Dr. Venkatesh.
Sure. And they know my email, so they can directly contact me also if they want any clarification or.
Definitely. So we'll be sharing both you and Dr. Karthik's email address with them, and if there are any questions which they were not answered because of any technical glitches, they could probably get in touch with you directly. And we'll also be sharing the video of this if there are. So there's one more question just came up. So it's as with Durgesh was asking, is there any AI-based software or tool for detecting coronavirus?
There are quite a few. I think the Chinese themselves have developed. So there are quite a few tools which they've taken a lot of data and then try to get input-output tests. And I think there is one for even who will. I don't know about detection, but probably I think they have for the ones who will give severe response. The sepsis kind of a response, which patient will give severe response. Okay, so for that, I think there is a platform, AIML platform. So after you're infected, based on some inputs in terms of oxygen dynamics, CO2 dynamics, and some immune markers, so you can predict whether they'll go into a shock or not. Okay, immune shock or not. So I think those kinds of platforms are there. I don't know about detection.
Okay. So thanks a lot, Dr. Venkatesh, for such an interesting session, and Dr. Karthik also. Unfortunately, I think he's not joined again. But thanks a lot, Dr. Karthik and Dr. Venkatesh, for such an engaging session, and thank you all the participants for making this very interactive and asking very interesting questions. And hope we plan to have a lot of similar webinars every month for the next several months we have planned. And hopefully we'll see all the participants again for the next webinar also. And thank you again, Dr. Venkatesh. And
yeah, okay, bye-bye. Yeah, thanks a lot.
Thank you. Yeah.
Surat?
Yeah. Okay. So I can just should I stop recording this? Okay.
No, I'll end it. That's okay.
Okay. Okay. Fine. I'll just call you.